Hello, my name is Nicholas Chia, and I'm pleased to be presenting to you today on machine learning approaches for modeling complex community dynamics. Now, this touches upon one of my favorite topics, which is modeling complex community dynamics, which is thinking about how, especially in the microbiome world, a milieu of different species get together to perform important tasks that affect our health or promote disease. Now, this has classically been a task that is essentially too hard for the human mind to encompass all of that complexity at once. But over the last decade or so, there's been a growth in machine learning approaches that really augment our ability to embrace that complexity and understand what's going on in the complex ecosystem that is the human body. So let's start with a non-human body first. Here's the Tesla bot. You may have heard of it. This is Elon Musk's dream that we're all going to own a Tesla bot, and it's going to help us go perform some mundane tasks like shopping for groceries. It's also going to help us perform mundane sort of mental tasks and physical tasks like looking at a bunch of apples and saying, well, this one's the bad one, so I'm going to throw it out and I'm only going to keep the good ones. And that's pretty much been the story of artificial intelligence and machine learning up until now. We've really talked about mostly scaling up mundane tasks or some standard classification and sorting algorithms where we're trying to sort class A from class B. And that's true in the microbiome world as well. You could think of a random forest as a mundane task of setting up many decision trees, and then using those to classify disease from health. Like in this example, where we're looking at the role of BMI and obesity in colorectal cancer. Other examples of classification come from the deep neural network world, deep learning, which has touched many fields. And it's been used in examples like this one for drug response, where Dr. Sung's lab has published a paper looking at how predictive the microbiome is for RA drugs. And it turns out the microbiome is the most predictive feature out of the mixture of features, clinical and microbiome and metabolomic. And these are some of the reasons why machine learning approaches have become so important in the microbiome world. Now, that being said, if you just look at the headlines, you know, they're review articles just within the last year or so, and I just pulled these out. These are not representative of everything. But if you look at all of the review articles talking about machine learning and the microbiome, you will see almost all of them fall into this category of basic classification and sorting. Well, you know, as someone with a quantitative background, I have to say that these tasks are not really satisfying. I mean, these are all just about dividing data up into different spaces using deep neural networks, finding, you know, the line that separates data points. And that's a very important task for biomarkers, but it's not as far as we could go with machine learning. It's not, to me, using the best use, making the best use of advances in machine learning that we have today. So this is really not a very satisfying feeling because, you know, my feeling is that we really want to gain insight out of these systems. We want to understand what the causal drivers are. We want to understand what the underlying processes are that give rise to disease and health. And biomarkers are good for prediction in certain things, but they're not helping us understand the science, which as an academic is really what gives you that satisfying feeling of being able to move the world forward. So the question I'm going to pose today is, can we use machine learning to improve microbiome modeling? And so examples of modeling community dynamics come in many forms. One example is from the Lotka-Volterra case over here, which is a very simple case many of you have seen before. It basically involves species-species interactions. And so if two species help each other grow, they have a positive interaction. This beta term becomes I guess a positive, well, I guess in this case it would be a negative term to make this positive growth-wise, or vice versa. If they hurt each other, they really have a negative term, and so on. Conway's Game of Life, which is really talking about spatial interactions. Conway's Game of Life is a game where, depending on how many neighbors you have different birth and death processes. If you're all alone, you die. If you're sort of next to a few people, you give birth. And if you're, have too many people, you overpopulate and people die. And this shows you that even with simple spatial interactions, you can get sort of these complex patterns. And then lastly, metabolic modeling, which is an area that I've personally dabbled in and enjoy. And it really has to do with how can we turn the biochemical constraints that we know, the biological knowledge, and put that all together into a giant understanding of reaction fluxes and use constraint-based flux balance approaches to make pointed predictions. And this has been used for community dynamics as well on a limited basis. And so this is a picture of John Conway. And the reason I put this picture here is to say that these are all limited by the human mind, these approaches, all of them. We really are having trouble scaling these up because we are very, very finite beings. And, we don't communicate, oddly enough, as well as computers do with each other. And so we don't scale up in the same way computers can. So how can we scale up these complex models? So first, let's go over what a complex model is. Fundamental definition of a complex model is that it has lots and lots of members and it's strongly interacting. So I say that so that we all agree that in order to understand a complex model, we are going to need to scale up. And personally, I think they're very fun because they're not very straightforward. They involve a little bit of convolutedness in answering them and complexity. But for people who just want the answer to the biology as in why this happens, they're a little bit painful, right? I mean, in some sense, the reason why we are going after biomarkers for medicine is because, you don't want your doctor to really wonder about your diagnosis so much. You really want them to just give you a diagnosis and use one marker and say, that's it, I know the answer. But that's not really realistic. Because in a complex system, like most biological systems, the interactions and not the members determine the behavior of the system. In other words, the interactions become stronger than the nature of the members themselves. And so when you're looking at an individual biomarker, you're really looking at one entity in the system, right? One type of agent, one species. But you're not looking at the interactions. And I will put out that the interactions are more important. And these interactions often give rise to collective effects that create seemingly ordered behavior. And that's exactly the kind of thing that we want to understand, right? What makes for certain types of gut bacteria configurations? What creates a healthy system? What does dysbiotic mean? All these sorts of questions that we want to know. And one example of this collective behavior is the formation of biofilms. Microbes do this all the time. They start growing as individual cells, they form larger bodies. Eventually they form these complex structures and even fruiting bodies that go off and then seed other things. And so without these collective effects, we wouldn't be able to understand even a biofilm of a single species, forget about the collective dynamics of many, many species. And this is something that I modeled once in the past, so this example wasn't pulled out of nowhere. And I have shown that we can model the formation of a biofilm using a set of master equations similar to the Lotka-Volterra equations. So going back to Lotka-Volterra, how are we going to scale this up? Well, one way that we looked at was taking Lotka-Volterra, putting out the generalized Lotka-Volterra equation, where this is the rate of change of a species xi. This is the rate of growth. This is the carrying capacity, K over here. And here, these alpha ij's specify the species-species interactions. And what we managed to show using data from Jeff Gore's lab that was published previously is that even for a three-way system, three interactions, the Lotka-Volterra model had a lot of trouble accurately depicting these relationships. So based on the pairwise interaction. So that's to say that if an experiment was done with just two species and each pair was simulated together, then putting all three of them together with the same interaction term did a really, really poor job. And these plots down here are showing the deviation from what we would expect based on the pairwise interaction terms. So this kind of showed us that this didn't work very well overall. And we tested this for larger systems. And we tested this by just looking at, well, what happens if we just simulate each species individually, just looking at the carrying capacity? What happens if we fit based on the pairwise growth data, where Gore's lab did this wonderful work where they actually grew up each pair and each organism individually, and then they compared growth rates in a community and outside of a community. And what we find is that these data points don't fit the data very well when we want to put together a whole system. And if we were to fit the whole system naively, we could do a much, much better job of it, although we still have some problems. So the issue with this is, of course, this fundamental model is not quite working and is not quite scaling up very well. So, and we can see that because in these three different configurations, we get very, very different parameter values overall. This is showing the percentage of parameter values that stays the same across these three circumstances. So pairwise growth parameters don't inform a community Lotka-Volterra model very well, making this particular model not particularly good in its formulation for scaling up. Yet we all love the Lotka-Volterra model because mentally we can understand what a species-species interaction is. But why is it failing, right? It's probably failing because we're missing some kind of interaction. So the Lotka-Volterra description is not sufficient. So what do we do? So what we did in our work was we looked at changing the form of the equations to account for more biology. In our case, because of prior experience with metabolic modeling, we said, why don't we try a metabolite-mediated model, which is to say that these microbes are not having direct species-species interactions, but they're interacting through the medium of some kind of metabolite. And that allows for more complex forms. And as you can see, we can now model the same thing, and we can add metabolites. And this becomes more complex, and it also allows us to have more fitting parameters. And so it's, of course, probably no surprise that that worked really well. But it also created a really large space of possibilities that, again, relies on human cleverness to sort of curate and understand how to search appropriately, right? Otherwise, the computer could fit based on any metabolite it really wanted to, but those might not be the real driver metabolites. So can we use machine learning to identify the new functional forms to describe complex systems based on limited time longitudinal data? That's the question I'm going to pose right now and bring forward and show you an example of. So the answer is yes, and I'm going to show you some work based on a picindy package that's actually remarkably easy to download and run that basically tries to guess what the functional forms are and tries to force those functional forms to be as sparse as possible, that is, to have as few terms as possible, which make it easier to understand from the human trying to interpret it on the other end. So we start with a system of equations. So let me go really slowly here. This is, again, on the left-hand side of the equation, the rate of change in X, and X can be a species, for instance, and there might be many X's, X1, X2, X3. There is then on the right-hand side a function describing the rate of change that depends on the value of X at a time t. So an example of that that you all might be familiar with by now is the SIR model that has been talked about in COVID-19 quite a bit, where you have a susceptible population whose rate in change over time is given in proportion to the number of infected people and the number of susceptible people they come in contact with. And that's very intuitive because the number of infected people can go on and infect more people. So the more infections you have, the more rampant it is in the community. But if there are no susceptible people, it's not going to spread anywhere. So you're not going to grow or lose in this case because the minus sign any of your susceptible people, right? So you need a mixture of both. Likewise, you have your infected people, and they continue to grow according to the same term because susceptible people become infected people, and then there's some exit rate for this system as well. So this is an example that you all might be familiar with, just so that we understand how this system of equations works. So when we're setting up this problem, what we can do using picindy is specify a number of functional forms, polynomial forms of the different species, x1, x2, so on, and they can be directly proportional. They can be proportional to the square, the cube, and so on. And we're going to give it a certain number of terms to search, and then it also can do these in combination as well, where it's you can have terms where species 1 is interacting with species 2, species 1 is interacting with species 1 and 2 and 3, and so on, in all different combinations with different powers up to a certain amount. And picindy says if we can make the number of non-zero terms sparse, then we can solve for the functional form quite well and very quickly. And as an example of that, we went back to the Lotka-Volterra because everybody likes it. And for 2 species Lotka-Volterra, we can set up our own equations, set the coefficients, and look at the true vector field and plot it, and then we can generate these 5 trajectories. Well, what we can do then is give those trajectories over to picindy, and we can have it impute the vector field over the whole space. And as you can see, it does really, really well. Just with 5 trajectories, it does a perfect job, essentially, of recapturing a low-order equation that describes the Lotka-Volterra vector field. So that's really good. And the question is, could we do it for something more complex? Well, if we want to consider a microbiome, we consider all the different absolute species and species abundances. And we can take this example from Stephanie's song and my collaborator in Wellesley, Mark Tuttle, where they gathered daily vaginal samples across the course of 11 weeks or so. And we can take each of these as a trajectory and as a discrete trajectory, and then we can use the same approach to try to understand the equations of motion, essentially, that describe the vector field of the microbiome, of the vaginal microbiome. There's the rate of change, you'll notice, in the abundance of species X. And I'll just note that this is approximated the difference between 2 subsequent time points. And when we do that and we plot this out, we get something very interesting, right? We get a very compelling result, which is that we can generate that vector field, then we can take a random point in the vector field and we can simulate forward. And we find that we get behavior that's very similar to what we see in real life. This is the relative abundance of L inners in blue. This is in crispatus in orange. And you see here from this example, you have this switching where this is purple, this is crispatus is purple, and blue is lactobacillus inners. And you can see that they switch in dominance very rapidly. And we have that same behavior captured in our model as well. In addition to that, the other thing that very often happens, in fact, in the same person, just in year 2 of this study, we have L crispatus dominance. And that dominance often does not, it fluctuates downward, but it really doesn't go anywhere across time. And here, we have similar behavior, although I'll note that there's a numerical instability and that this is preliminary data that we're still working through, but it's a compelling quantitative result that we've produced here. So one last thought here is that you can think of this rate of change also as the replication rate of species X. And for that, if you have metagenomics time longitudinal sampling, you actually can use one of these metagenomic growth rate estimators. There are a few of them around. 2 examples are sort of shown here. And if you have that, you could perhaps replace this side and actually improve your estimate even further. I, of course, am not going to show an example of that because I don't have that data. But if anybody does, feel free to email me if you would like to collaborate and give that a try. All right. So that's one way in which we can use machine learning to improve these sorts of understanding approaches, right? Approaches to understanding biology. Another one I'd like to talk about is in evolution. And the particular example we looked at in this case was cancer evolution, but this could be applied to any population, including microbial populations. And for those of you unfamiliar with cancer, one of the paradigms of cancer is that it starts with a mutation, That mutation grows because it creates a proliferative advantage. More mutations accumulate, and so on. And that's the process of cancer evolution until you get to, a tumor that causes health problems. Now we can simulate that by considering genes in a genome. Those genes can have a value of 0 or 1 in our simulation, which is to say 0 is normal and 1 is mutated. And just for simplicity, we can start with a genome with only 4 gene loci, and there are only 2 driver loci at the very end, at the beginning and end of that genome. And so that means the cancer genomes are given by these 4 genomes over here that have a 1 in the first position and a 1 in the last position. And the non-cancer genomes are all the ones that have a 0 and one of those positions or both. And so we can simulate this forward now. That is, we can simulate the evolution of cancer. We can mark down the progression of that cancer, and then we can use those to try to understand what's happening. Now, how are we going to use those? Well, we can use a technique called inverse reinforcement learning. And I don't want to dive too deep into this, but inverse reinforcement learning basically takes a set of expert demonstrations, which is to say demonstrations of cancer evolution, and it infers a reward function that describes the incentives and preferences that that agent is going to behave with. That is to say, it's understanding how cancer behaves. It's forming the reward map of cancer in this case. And from our example, I already told you that the first and last, because we put this in, we knew what the answer was. I told you that the first and last position were the positions that were driver mutations. And what you can see in this simulation is that indeed that's true. The first position and the last position, if they're both 0, are approximately equally rewarded in this reward map in terms of these actions, which are mutating the first or the last position. If the first position is already mutated, the last position is what really wants to be picked up. If it's the opposite, the last position is mutated, then the first position is the one that's the most rewarding. And so you could see that this reward function describes the behaviors that would drive cancer. And so this becomes a really interesting paradigm for understanding cancer evolution. And the question we ask is, can we learn what incentivizes tumor growth? Can we take the behavior of cancer, that is the expert demonstrations that come from cancer, which we're going to estimate using tumor phylogeny, and recover meaningful reward functions that describe tumor growth? So for this, we created an algorithm which we called the pop-up restaurant for inverse reinforcement learning, or pure IRL. The reason we call it the pop-up restaurant process is because it involves essentially a lot of Chinese restaurant processes for partitioning expert demonstrations into different reward functions. And because the name matches quite well. A pop-up restaurant is a short-term restaurant that allows chefs to try out new concepts, new audiences, and experiment with new ingredients. And that's pretty much what we're doing. We're trying new reward functions, reshuffling the expert demonstrations into different restaurants, and trying different types of data in all of these until we find sort of the best result. And again, not going to go into the details of this, but truth in advertising, this is the pure IRL implementation, and anyone who wants to read this is welcome to go through our paper, which I'll show at the end of the talk. So the result of this is that we showed that the 2 most rewarded paths, the 2 most likely paths that occur after learning the reward functions, are these 2 shown here. This is sort of the reward. This is the reward function up here, although it's really hard to read because you can see this space is really, really large. What I can say about this space is that there are about 10 to the 200 possible places you can go here, or different orders in which you can do things. And the top 2 give us these orderings, APC, KRAS, maybe SMAD4, TP53, leading to cancer. And what's really interesting is this exactly matches, the second path exactly matches the 1990 seminal paper by Fearson and Vogelstein, which described the progression of colorectal cancer that is still considered canonical today. So in other words, we managed to rediscover this path and we only used data from 27 patients with no time longitudinal data, just inferring based on tumor phylogeny, which gives us only a very approximate sense of time. So this really shows us the power of this method, and it has the potential to add, it doesn't just rediscover what humans already know, it has the potential to discover new things. And so some of these new things, we looked at some of the more likely transitions from normal that were considered drivers in this model. And we found these papers for all three of these. We instantly found papers that indicated either experimental evidence or epidemiological evidence from meta studies that show these are early drivers in colorectal cancer. So this is really a new paradigm for us pushing forward to use the computer to augment biological understanding. So in conclusion, I want us all to understand that biological systems are really complex and have complex behavior. Machine learning is a way in which we can embrace that. And especially exciting are the growth avenues that involve understanding the infinite dynamics, such as those infinite dynamics that include evolution or microbiome behavior, ecological behavior. And those are the areas that are really exciting and upcoming in the future for the microbiome field. Here are a bunch of papers for future reading, and I want to give thanks to everyone in the lab for this and my collaborators, my funding sources. I'll highlight a few people in particular here. John Kalantari, who now runs his own lab and group within Mayo Clinic, was the postdoc who did the work on inverse reinforcement learning. Björn Brunner did the work on the ecological models and the metabolite-mediated models and is now moving on to postdoc in Los Alamos National Lab. Stephanie Song, who I mentioned earlier, provided the vaginal microbiome data for us to work with. And Yash Lal actually did the work with the vector fields that I showed in the middle of this talk. So thank you very much. And with that, I hope you all had a good time.